Thymus histology ChatGPT. Thymus histology refers to the microscopic structure and organization of the thymus gland, a primary lymphoid organ crucial for the development and maturation of T lymphocytes T cells. Here's an overview of the histological features of the thymus. Structure of the thymus, capsule. The thymus is surrounded by a thin connective tissue capsule that extends inward, dividing the gland into lobules. Cortex. Outer region. The cortex is the outer part of each thymic lobule and appears dark under a microscope. Thymic capsules, corpuscles, concentric whorls of epithelial cells found in the cortex, which are involved in regulatory functions within the thymus. Medulla. Inner region. The medulla is located centrally within each thymic lobule and appears lighter in color compared to the cortex. Thymic medullary capsules, corpuscles, larger and more irregularly shaped compared to those in the cortex, often surrounded by lymphocytes. Cortico-medullary junction. The transition zone between the cortex and medulla, where developing T cells migrate from the cortex to the medulla for further maturation and selection. Cell types in thymus histology. Thymic epithelial cells TECs. Found throughout the thymus, TECs play a crucial role in supporting and guiding the maturation of T cells. Cortex. TECs in the cortex contribute to the positive selection of T cells. Medulla. TECs in the medulla contribute to the negative selection of T cells, removing those that may be self-reactive. T lymphocytes T cells. The primary cells of the thymus, derived from hematopoietic stem cells. Cortex. Developing T cells thymocytes undergo positive selection based on their ability to recognize self-major histocompatibility complex MHC molecules presented by TECs. Medulla. T cells that pass positive selection migrate to the medulla for further maturation and negative selection, ensuring self-tolerance and removing autoreactive T cells. Dendritic cells and macrophages. Present in both cortex and medulla, these cells participate in antigen presentation and phagocytosis of apoptotic cells during T cell maturation and selection processes. Functions of the thymus. T cell maturation. The thymus is crucial for the development and maturation of T cells, which play a central role in adaptive immunity. Tolerance induction. Through the processes of positive and negative selection, the thymus ensures that mature T cells are capable of recognizing foreign antigens while tolerating self-antigens, thereby preventing autoimmune responses. Histological techniques used. Hematoxylin and eosin H&E staining, used to visualize the overall structure of the thymus and identify different cell types. Immunohistochemistry IHC, utilized to identify specific cell markers, e.g., cytokeratins for TECs, CD markers for T cells within thymic tissue sections. Electron microscopy provides ultrastructural details of cell-cell interactions and morphological features within the thymus. Understanding thymus histology is crucial for studying immune system development, autoimmune diseases, and immunodeficiencies, offering insights into how T cell function is regulated and maintained throughout life. Structure and function and location of thymus ChatGPT. Structure of the thymus, location. The thymus gland is located in the upper anterior thoracic cavity, behind the sternum and between the lungs. It is situated above the heart and below the thyroid gland. Structure. The thymus gland is bilobed and has a pinkish gray color, with each lobe composed of lobules divided by connective tissue septa. Histological zones. Cortex, the outer region of each thymic lobule, containing densely packed immature T cells, thymocytes, and scattered thymic epithelial cells TECs. The cortex is involved in positive selection of T cells. Medulla, the inner region of each lobule, characterized by fewer thymocytes and a higher concentration of TECs, dendritic cells, and macrophages. The medulla is involved in negative selection and tolerance induction of T cells. Thymic capsules, corpuscles. Found in the medulla, these are concentric whorls of epithelial cells that are involved in regulatory functions within the thymus, although their exact role is still not fully understood. Function of the thymus. T cell maturation. The primary function of the thymus is to support the maturation and differentiation of T lymphocytes T cells, a type of white blood cell crucial for adaptive immunity. Positive selection. In the cortex, thymocytes that can recognize self-major histocompatibility complex MHC molecules presented by thymic epithelial cells TECs survive and mature. Negative selection. Negative selection. In the medulla, thymocytes that react strongly to self-antigens undergo apoptosis, cell death, preventing the development of T cells that could cause autoimmune reactions. Immune tolerance. Through the processes of positive and negative selection, the thymus ensures that mature T cells are capable of recognizing foreign antigens while tolerating self-antigens, thus preventing autoimmune diseases. Hormonal function. The thymus secretes several hormones, such as thymosin, thymolin, and thymopoietin, which are involved in the development and differentiation of T cells. Clinical significance. Thymectomy, surgical removal of the thymus, often performed in certain medical conditions, such as myasthenia gravis or thymic tumors, can affect immune function and T cell development. Thymic involution. The thymus undergoes progressive involution with age, leading to a decrease in its size and activity. This process contributes to age-related decline in immune function. Understanding the structure, function, and location of the thymus is crucial for comprehending its role in immune system development and regulation, as well as its implications in health and disease. Clinical significance of thymus in detail and diseases in short ChatGPT. The thymus plays a crucial role in immune system development and function, particularly in the maturation and regulation of T lymphocytes T cells. Its clinical significance extends to various aspects of health and disease. Functions of the thymus, T cell maturation. The thymus is the primary organ where T cells mature from precursor cells originating in the bone marrow. Through processes of positive and negative selection, T cells learn to recognize foreign antigens while avoiding self-reactivity. Immune tolerance. Negative selection in the thymus ensures that T cells do not recognize self-antigens, thus preventing autoimmune reactions and promoting immune tolerance. Hormone production. The thymus produces hormones such as thymosin, thymolin, and thymopoietin, which play roles in T cell differentiation and development. Clinical significance. Thymectomy. Surgical removal of the thymus. Thymectomy is sometimes necessary in medical conditions such as thymic tumors, myasthenia gravis, and other autoimmune disorders. Myasthenia gravis, a condition where autoantibodies attack acetylcholine receptors at neuromuscular junctions, causing muscle weakness. Thymectomy can improve symptoms in some patients. Thymic involution. With age, the thymus undergoes progressive involution, leading to a decrease in its size and activity. This process contributes to age-related decline in immune function and susceptibility to infections. Immunodeficiency disorders. Thymic abnormalities or dysfunction can lead to primary immunodeficiency disorders, where T cell development and function are impaired. DiGeorge syndrome, caused by a genetic defect affecting thymus development and resulting in T cell deficiency, leading to recurrent infections and other health issues. Autoimmune diseases. Disorders where the immune system mistakenly attacks the body's own tissues can be influenced by thymic function and T cell development. Autoimmune polyendocrine syndrome (APS) associated with autoimmune disorders affecting multiple endocrine glands, often linked to abnormalities in thymic function. Cancer. Thymic tumors, although rare, can develop within the thymus gland and may require surgical intervention or other treatments. Thymoma, a tumor originating from thymic epithelial cells, which can be benign or malignant. Aging and immune senescence. Thymic involution contributes to age-related decline in immune function, affecting T cell diversity and responsiveness. Summary. The thymus is integral to immune system development, tolerance induction, and response to infections and autoimmune diseases. Understanding its role in health and disease underscores its clinical significance in various medical conditions, from autoimmune disorders to immunodeficiencies and cancers. Bone marrow histology ChatGPT. Bone marrow histology refers to the microscopic structure and composition of bone marrow, the soft tissue found within the cavities of bones. Here's an overview of bone marrow histology, its components and functions. Types of bone marrow. Red bone marrow. Found in the cavities of flat bones, e.g., sternum, pelvis, ribs, and the epiphyses of long bones, e.g., femur, humerus. Responsible for hematopoiesis, the process of blood cell formation, including erythrocytes, red blood cells, leukocytes, white blood cells, and platelets. Yellow bone marrow. Predominantly found in the medullary cavity of long bones. Composed mainly of adipocytes, fat cells, and functions as a storehouse of fat. Structure of bone marrow. Cellular composition. Hematopoietic cells include hematopoietic stem cells, HSCs, and progenitor cells that give rise to all blood cell types. Stromal cells, supportive cells including fibroblasts, adipocytes, and macrophages. Endothelial cells form blood vessels that supply nutrients and oxygen to the bone marrow. Histological zones. Trabecular cancellous bone, meshwork of bony trabeculae that provides structural support. Hematopoietic parenchyma, composed of hematopoietic cells arranged in cords or islands surrounded by sinusoidal blood vessels. Sinusoidal endothelium, specialized endothelial cells lining sinusoidal blood vessels that facilitate the movement of blood cells into and out of the marrow. Bone marrow spaces. Red marrow spaces, rich in hematopoietic cells, with a network of sinusoidal capillaries facilitating the exchange of nutrients and waste products. Yellow marrow spaces, dominated by adipocytes, with fewer hematopoietic elements compared to red marrow. Functions of bone marrow. Hematopoiesis. Production and maturation of blood cells from hematopoietic stem cells, including erythrocytes, red blood cells, leukocytes, white blood cells, and platelets. Immune function. Bone marrow houses immune cells such as lymphocytes, B cells, and T cells, macrophages, and dendritic cells, contributing to immune surveillance and defense against pathogens. Storage and release. Red marrow stores and releases blood cells into the circulation as needed, maintaining homeostasis of blood cell populations. Yellow marrow serves as an energy reserve, providing adipocytes for energy storage. Clinical applications. Bone marrow aspiration and biopsy. Diagnostic procedures to assess hematopoietic cellularity, presence of abnormal cells, e.g., leukemia, and bone marrow function. Bone marrow transplantation. Used therapeutically to replace diseased or damaged bone marrow with healthy stem cells in conditions such as leukemia, lymphoma, and genetic disorders affecting blood cell production. Understanding bone marrow histology is essential for comprehending its role in hematopoiesis, immune function, and clinical applications in health and disease. Histological examination of bone marrow provides valuable insights into blood cell formation, disorders affecting blood cell production, and therapeutic interventions. Structure. Location ChatGPT. Certainly. Here's a detailed overview of the structure and location of bone marrow. Structure of bone marrow. Red bone marrow. Composition. Contains hematopoietic cells, including hematopoietic stem cells, HSCs, progenitor cells, and various blood cell types in different stages of maturation, erythrocytes, leukocytes, and platelets. Histological features. Appears dark red due to its high vascularity and is composed of a network of sinusoidal capillaries surrounded by islands or cords of hematopoietic cells. Yellow bone marrow. Composition. Predominantly consists of adipocytes, fat cells with sparse hematopoietic cells, especially in adults. Location. Found mainly in the medullary cavity of long bones and can also be present in the cavities of other bones. Location of bone marrow. Red bone marrow. Primary sites. Found in the cavities of flat bones, e.g., sternum, ribs, pelvis, and the epiphysis ends of long bones, e.g., femur, humerus. Distribution. Red marrow is more prevalent in children and young adults, gradually transitioning to yellow marrow with age as hematopoietic activity decreases. Yellow bone marrow. Primary sites. Occupies the central medullary cavity of long bones, such as the femur, tibia, and humerus. Role. Acts primarily as a site for fat storage and energy reserve, with a reduced capacity for hematopoiesis compared to red marrow. Functions of bone marrow. Hematopoiesis. Red bone marrow is responsible for the continuous production and maturation of blood cells throughout life, including erythrocytes, red blood cells, leukocytes, white blood cells, and platelets. Immune function. Bone marrow serves as a reservoir for immune cells, including lymphocytes, B cells, and T cells, macrophages, and dendritic cells, contributing to immune surveillance and defense against infections. Storage and release. Red marrow stores blood cells and releases them into the circulation as needed to maintain the body's blood cell populations and respond to physiological demands. Clinical relevance. Bone marrow aspiration and biopsy. Diagnostic procedures used to evaluate hematopoietic cellularity, assess the presence of abnormal cells, e.g., leukemia, and investigate bone marrow disorders. Bone marrow transplantation. Therapeutic procedure involving the infusion of healthy hematopoietic stem cells to replace damaged or diseased marrow in conditions such as leukemia, lymphoma, and certain genetic disorders. Understanding the structure and location of bone marrow is crucial for appreciating its role in hematopoiesis, immune function, and its clinical implications in health and disease management. Clinical significance and diseases ChatGPT. The clinical significance of bone marrow is profound, as it plays essential roles in hematopoiesis, immune function, and is a site for certain diseases and therapeutic interventions. Here's an overview of its clinical relevance and associated diseases. Clinical significance. Hematopoiesis. Bone marrow is crucial for the production and maturation of blood cells, including erythrocytes, red blood cells, leukocytes, white blood cells, and platelets. Disorders affecting bone marrow can lead to abnormalities in blood cell production, resulting in anemia, leukopenia, thrombocytopenia, and other hematologic conditions. Immune function. Bone marrow houses immune cells such as lymphocytes, B cells, and T cells, macrophages, and dendritic cells, contributing to immune surveillance, response to infections, and autoimmune diseases. Diagnostic procedures. Bone marrow aspiration and biopsy, used to diagnose and stage various hematologic disorders, including leukemia, lymphoma, myelodysplastic syndromes, and bone marrow metastasis from solid tumors. Flow cytometry analyzes bone marrow cells to identify and characterize abnormal populations, aiding in the diagnosis of blood cancers and immunodeficiencies. Therapeutic interventions. Bone marrow transplantation (BMT), also known as hematopoietic stem cell transplantation (HSCT-BMT), is a treatment for hematologic malignancies, e.g., leukemia, lymphoma, bone marrow failure syndromes, e.g., aplastic anemia, genetic disorders affecting blood cells, e.g., thalassemia, and certain immune deficiencies. Diseases and disorders involving bone marrow. Leukemia. Cancer of the blood or bone marrow, characterized by abnormal proliferation of immature white blood cells, leukocytes. Types include acute myeloid leukemia (AML), acute lymphoblastic leukemia, all chronic myeloid leukemia (CML), and chronic lymphocytic leukemia (KA). Lymphoma. Cancer originating in lymphocytes, which may involve bone marrow infiltration. Types include Hodgkin lymphoma and non-Hodgkin lymphoma. Myeloproliferative disorders. Conditions where bone marrow produces excessive numbers of mature blood cells. Examples include polycythemia vera, essential thrombocythemia, and myelofibrosis. Myelodysplastic syndromes (MDS). Group of disorders characterized by ineffective production of blood cells due to abnormal hematopoietic stem cell function. MDS can progress to acute leukemia, aplastic anemia. Failure of bone marrow to produce adequate numbers of blood cells, leading to pancytopenia, low counts of all blood cell types. Multiple myeloma. Cancer of plasma cells, a type of white blood cell that produces antibodies. It often involves bone marrow infiltration and can lead to bone destruction and marrow failure. Metastatic bone marrow infiltration. Spread of cancer from other organs e.g., breast, lung, prostate to the bone marrow, impairing normal hematopoiesis and causing complications such as anemia and thrombocytopenia. Bone marrow failure syndromes. Conditions characterized by inadequate production of blood cells, leading to cytopenias, deficiencies in one or more blood cell types. Examples include Fanconi anemia and Diamond-Blackfan anemia. Understanding the clinical significance of bone marrow helps in the diagnosis, treatment, and management of various hematologic disorders and cancers. Advances in bone marrow transplantation and supportive care have significantly improved outcomes for patients with these conditions, highlighting the critical role of bone marrow in hematopoiesis and immune function. Lymph nodes histology ChatGPT. Lymph nodes are an integral part of the lymphatic system, serving as important sites for immune surveillance and response. Here's an overview of lymph node histology, detailing their structure and functions. Structure of lymph nodes, capsule. Lymph nodes are surrounded by a fibrous capsule that extends inward, forming trabeculae that divide the node into compartments. Cortex. Outer cortex, superficial cortex contains densely packed lymphoid nodules, follicles that house B cells and are primary sites of antigen presentation. Paracortex lies between the cortex and medulla, containing T cells and high endothelial venules, HEVs, through which lymphocytes enter the node. Medulla. Medullary cords. Outer cortex, superficial cortex contains densely packed lymphoid nodules, follicles that house B cells and are primary sites of antigen presentation. Paracortex lies between the cortex and medulla, containing T cells and high endothelial venules, HEVs, through which lymphocytes enter the node. Medulla. Medullary cords extend inward from the cortex, containing plasma cells, macrophages, and lymphocytes. Medullary sinuses, spaces between the medullary cords that drain lymph into efferent lymphatic vessels. Circulation and drainage. Afferent lymphatic vessels bring lymph into the lymph node, where it circulates through the cortex and medulla before exiting via efferent lymphatic vessels. Lymphocyte circulation allows for immune surveillance and activation in response to antigens presented within the node. Functions of lymph nodes. Filtration and immune response. Lymph nodes filter lymphatic fluid, removing foreign particles, antigens, and pathogens. Immune cells within the nodes, such as lymphocytes and macrophages, respond to these antigens, initiating immune responses. Antigen presentation. Dendritic cells and macrophages in the cortex and paracortex capture antigens from lymph and present them to T cells, initiating adaptive immune responses. B cell activation and antibody production. B cells in the follicles of the cortex can be activated by antigen-presenting cells (APCs) and differentiate into plasma cells that produce antibodies against specific antigens. Histological techniques used. Hematoxylin and eosin H&E staining provides general structural details, highlighting different regions such as the cortex, paracortex, and medulla. Immunohistochemistry (IHC) utilized to identify specific cell types, e.g., B cells, T cells, macrophages, or molecules, e.g., cytokines, antigens within lymph node tissue sections. Electron microscopy offers ultrastructural insights into cellular interactions, particularly between lymphocytes, dendritic cells, and antigens. Clinical relevance. Lymphadenopathy, enlarged lymph nodes due to infections, e.g., bacterial, viral inflammation, autoimmune disorders, or malignancies. Lymph node biopsy, diagnostic procedure to investigate the cause of lymphadenopathy, determine lymph node involvement in cancers, e.g., lymphoma, metastatic tumors, and assess immune responses. Sentinel lymph node biopsy, technique used in cancer staging to identify the first lymph nodes draining a tumor site, crucial for determining cancer spread and treatment planning. Understanding lymph node histology provides insights into their roles in immune function, response to infections, and their clinical implications in health and disease. Understanding lymph node histology provides insights into their roles in immune function, response to infections, and their clinical implications in health and disease. Lymph node structure and location functions ChatGPT. Structure of lymph nodes, capsule. Lymph nodes are surrounded by a fibrous capsule that extends inward, forming trabeculae that divide the node into compartments. Cortex. Outer cortex, superficial cortex contains densely packed lymphoid nodules, follicles that house B cells and are primary sites of antigen presentation. Paracortex lies between the cortex and medulla, containing T cells and high endothelial venules, HEVs, through which lymphocytes enter the node. Medulla. Medullary cords extend inward from the cortex, containing outer cortex, superficial cortex contains densely packed lymphoid nodules, follicles that house B cells and are primary sites of antigen presentation. Paracortex lies between the cortex and medulla, containing T cells and high endothelial venules, HEVs, through which lymphocytes enter the node. Medulla. Medullary cords extend inward from the cortex, containing plasma cells, macrophages, and lymphocytes. Medullary sinuses, spaces between the medullary cords that drain lymph into efferent lymphatic vessels. Circulation and drainage. Afferent lymphatic vessels bring lymph into the lymph node, where it circulates through the cortex and medulla before exiting via efferent lymphatic vessels. Lymphocyte circulation allows for immune surveillance and activation in response to antigens presented within the node. Location of lymph nodes. Lymph nodes are distributed throughout the body along lymphatic vessels. They are concentrated in areas where lymphatic drainage is significant, such as cervical region, neck area, including submandibular, cervical, and supraclavicular lymph nodes. Axillary region, armpit area, draining lymph from the upper limbs, chest wall, and breast. Inguinal region, groin area, receiving lymph from the lower limbs and pelvic organs. Mesenteric region, abdominal area, associated with lymph drainage from the gastrointestinal tract. Pelvic region, includes iliac and pelvic lymph nodes, draining lymph from pelvic organs. Functions of lymph nodes. Filtration and immune response. Lymph nodes filter lymphatic fluid, removing foreign particles, antigens, and pathogens. Immune cells within the nodes, such as lymphocytes and macrophages, respond to these antigens, initiating immune responses. Antigen presentation. Dendritic cells and macrophages in the cortex and paracortex capture antigens from lymph and present them to T cells, initiating adaptive immune responses. B cell activation and antibody production. B cells in the follicles of the cortex can be activated by antigen-presenting cells (APCs) and differentiate into plasma cells that produce antibodies against specific antigens. Immune surveillance. Lymph nodes serve as checkpoints where immune cells monitor lymphatic fluid for potential threats, helping to detect and respond to infections and abnormal cells. Clinical relevance. Lymphadenopathy, enlarged lymph nodes due to infections, e.g., bacterial, viral inflammation, autoimmune disorders, or malignancies. Lymph node biopsy, diagnostic procedure to investigate the cause of lymphadenopathy, determine lymph node involvement in cancers, e.g., lymphoma, metastatic tumors, and assess immune responses. Sentinel lymph node biopsy, technique used in cancer staging to identify the first lymph nodes draining a tumor site, crucial for determining cancer spread and treatment planning. Understanding the structure, location, and functions of lymph nodes is essential for comprehending their role in immune function, response to infections, and their clinical significance in diagnosing and managing various diseases. Clinical significance in diseases ChatGPT. The clinical significance of lymph nodes is profound, as they play crucial roles in immune function, disease detection, and as indicators of various health conditions. Here's an overview of their clinical relevance and associated diseases. Clinical significance, immune function. Lymph nodes are key organs in the immune system, where immune cells such as lymphocytes, macrophages, and dendritic cells interact to mount immune responses against pathogens, antigens, and abnormal cells. Disease detection and diagnosis. Lymphadenopathy, enlarged lymph nodes, palpable on physical examination, may indicate underlying infections e.g., bacterial, viral, inflammatory conditions e.g., autoimmune diseases or malignancies. Lymph node biopsy, a diagnostic procedure involving the removal and examination of lymph node tissue to identify the cause of lymphadenopathy, assess immune responses, or stage cancers. Cancer staging and prognosis. Metastasis, cancer cells can spread to regional lymph nodes via lymphatic vessels. Sentinel lymph node biopsy is often used to clinical significance and diseases. ChatGPT. The clinical significance of lymph nodes is profound, as they play crucial roles in immune function, disease detection, and as indicators of various health conditions. Here's an overview of their clinical relevance and associated diseases. Clinical significance, immune function. Lymph nodes are key organs in the immune system, where immune cells such as lymphocytes, macrophages, and dendritic cells interact to mount immune responses against pathogens, antigens, and abnormal cells. Disease detection and diagnosis. Lymphadenopathy, enlarged lymph nodes, palpable on physical examination, may indicate underlying infections e.g., bacterial, viral, inflammatory conditions e.g., autoimmune diseases or malignancies. Lymph node biopsy, a diagnostic procedure involving the removal and examination of lymph node tissue to identify the cause of lymphadenopathy, assess immune responses, or stage cancers. Cancer staging and prognosis. Metastasis, cancer cells can spread to regional lymph nodes via lymphatic vessels. Sentinel lymph node biopsy is often used to determine if cancer has spread beyond its primary site, influencing treatment decisions and prognosis. Infections and inflammatory conditions. Lymph nodes enlarge in response to infections, acting as sites where immune cells combat pathogens and initiate immune responses. Persistent lymphadenopathy can indicate chronic infections or inflammatory diseases. Diseases involving lymph nodes. Lymphadenitis. Inflammation of lymph nodes due to bacterial or viral infections, causing localized swelling, tenderness, and sometimes abscess formation e.g., bacterial lymphadenitis. Lymphoma. Cancer of the lymphatic system, including Hodgkin lymphoma and non-Hodgkin lymphoma. Lymphoma can originate within lymph nodes or involve them through metastasis, impacting lymph node size, texture, and function. Metastatic cancer. Solid tumors e.g., breast, lung, melanoma can metastasize to regional lymph nodes via lymphatic vessels, altering their appearance and potentially causing pain or dysfunction. Autoimmune diseases. Conditions such as systemic lupus erythematosus (SLE) and rheumatoid arthritis can lead to lymphadenopathy due to chronic immune activation and inflammation affecting lymph node architecture and function. HIV/AIDS. Human immunodeficiency virus (HIV) infection can cause generalized lymphadenopathy as the virus targets and replicates within lymphoid tissues, impacting immune cell function and contributing to opportunistic infections. Diagnostic and therapeutic interventions. Lymph node biopsy, used to obtain tissue samples for histopathological examination, aiding in the diagnosis of infections, lymphomas, and metastatic cancers. Sentinel lymph node biopsy, technique to identify the first lymph nodes draining a tumor site, crucial for staging cancers and determining the need for further treatment e.g., lymphadenectomy, chemotherapy. Lymph node dissection, surgical removal of affected lymph nodes, performed in cases of extensive cancer spread or therapeutic intervention in lymphatic diseases. Understanding the clinical significance of lymph nodes helps healthcare providers in diagnosing diseases, planning treatments, and monitoring patient outcomes, highlighting their pivotal role in immune surveillance and disease management. Spleen histology. ChatGPT. The spleen is an important organ in the lymphatic system and plays various roles in immune function, blood filtration, and hematopoiesis. Here's an overview of spleen histology, detailing its structure and functions. Structure of the spleen, capsule. The spleen is surrounded by a dense fibrous capsule that extends inward as trabeculae, dividing the organ into compartments. Red pulp. Comprises about 75% of the spleen and is involved in filtering and processing blood. Splenic sinuses, large venous channels lined with macrophages, splenic sinusoidal cells that filter blood and remove older damaged red blood cells, erythrocytes. Splenic cords, cords of Billroth, meshwork of reticular fibers and macrophages that support blood flow and contain immune cells. White pulp. Concentrated around arterioles and forms clusters called periarteriolar lymphoid sheaths, pals, and lymphoid follicles. White pulp nodules. Pals, contains T lymphocytes involved in immune responses. Lymphoid follicles, contain B lymphocytes, which proliferate and differentiate into plasma cells upon antigen stimulation, producing. The spleen is surrounded by a dense fibrous capsule that. The spleen is an important organ in the lymphatic system and plays various roles in immune function, blood filtration, and hematopoiesis. Here's an overview of spleen histology, detailing its structure and functions. Structure of the spleen, capsule. The spleen is surrounded by a dense fibrous capsule that extends inward as trabeculae, dividing the organ into compartments. Red pulp. Comprises about 75% of the spleen and is involved in filtering and processing blood. Splenic sinuses, large venous channels lined with macrophages, splenic sinusoidal cells that filter blood and remove older damaged red blood cells, erythrocytes. Splenic cords, cords of Billroth, meshwork of reticular fibers and macrophages that support blood flow and contain immune cells. White pulp. Concentrated around arterioles and forms clusters called periarteriolar lymphoid sheaths, pals, and lymphoid follicles. White pulp nodules. Pals, contains T lymphocytes involved in immune responses. Lymphoid follicles, contain B lymphocytes, which proliferate and differentiate into plasma cells upon antigen stimulation, producing antibodies. Marginal zone. Separates the red pulp from the white pulp and contains specialized macrophages and lymphocytes that capture and process antigens from the blood. Functions of the spleen. Immune function. The spleen acts as a filter for blood, removing pathogens, antigens, and old damaged red blood cells. It also plays a role in immune surveillance in response to bloodborne infections. Hematopoiesis. During fetal development and certain pathological conditions e.g., anemia, the spleen can serve as a site of hematopoiesis, producing red blood cells, white blood cells, and platelets. Blood filtration. Macrophages in the red pulp phagocytose and destroy older abnormal red blood cells, recycling iron and other components for reuse. Storage. The spleen serves as a reservoir for blood, particularly platelets, which can be released into circulation in response to bleeding or other physiological demands. Clinical significance. Splenomegaly, enlargement of the spleen due to various causes, including infections e.g., mononucleosis, liver diseases e.g., cirrhosis, hematologic disorders e.g., leukemia, and inflammatory conditions e.g., autoimmune diseases. Splenectomy, surgical removal of the spleen, typically performed to treat conditions such as traumatic rupture, certain hematologic disorders e.g., immune thrombocytopenia, and splenic tumors. However, splenectomy increases the risk of infections, particularly those caused by encapsulated bacteria, due to loss of the splene's immune functions. Splenosis, the autotransplantation of splenic tissue into other parts of the body, often occurring after splenectomy, which may retain some immune functions. Understanding spleen histology and its functions is crucial for comprehending its roles in immune responses, blood filtration, and its clinical implications in health and disease. Structure, function, and location. ChatGPT. Structure of the spleen, capsule. The spleen is enveloped by a fibrous capsule that extends inward to form trabeculae, which divide the spleen into compartments. Red pulp makes up the majority of the spleen and is involved in filtering blood. Splenic sinusoids, large venous channels lined with macrophages, sinusoidal lining cells that filter blood and remove old or damaged red blood cells, erythrocytes. Splenic cords, cords of Billroth composed of reticular fibers and macrophages that support blood flow and contain immune cells. White pulp concentrated around arterioles and consists of periarteriolar lymphoid sheaths, pals, and lymphoid follicles. White pulp nodules. Pals contains T lymphocytes that participate in immune responses. Lymphoid follicles contain B lymphocytes that proliferate and differentiate into plasma cells upon antigen stimulation, producing antibodies. Marginal zone separates the red pulp from the white pulp and contains specialized macrophages and lymphocytes that capture and process antigens from the blood. Functions of the spleen. Immune function. Acts as a filter for blood, removing pathogens, antigens, and old damaged red blood cells. It plays a crucial role in immune surveillance in response to blood-borne infections. Hematopoiesis. During fetal development and in certain pathological conditions, e.g., anemia, the spleen can serve as a site of hematopoiesis, producing red blood cells, white blood cells, and platelets. Blood filtration. Macrophages in the red pulp phagocytose and degrade old or abnormal red blood cells, recycling iron and other components for reuse. Storage. Acts as a reservoir for blood, particularly platelets, which can be released into circulation in response to bleeding or other physiological demands. Location of the spleen. Located in the upper left quadrant of the abdomen, under the diaphragm and behind the stomach. Attached to the stomach by the gastrosplenic ligament and to the left kidney by the splenorenal ligament. The spleen's position allows it to interact with blood from the splenic artery and drain into the splenic vein, facilitating its filtration and immune functions. Clinical significance. Splenomegaly, enlargement of the spleen due to various causes, including infections e.g., mononucleosis, liver diseases e.g., cirrhosis, hematologic disorders e.g., leukemia, and inflammatory conditions e.g., autoimmune diseases. Splenectomy, surgical removal of the spleen, typically performed to treat conditions such as traumatic rupture, certain hematologic disorders e.g., immune thrombocytopenia, and splenic tumors. However, splenectomy increases the risk of infections, particularly those caused by encapsulated bacteria, due to loss of the spleen's immune functions. Splenosis, the autotransplantation of splenic tissue into other parts of the body, often occurring after splenectomy, which may retain some immune functions. Understanding the structure, functions, and location of the spleen is essential for comprehending its roles in immune responses, blood filtration, and its clinical implications in health and disease management. Clinical significance in diseases. ChatGPT. The spleen plays a crucial role in the immune system and hematologic function, and its clinical significance spans various diseases and conditions. Here's an overview of its clinical relevance and associated disorders. Clinical significance, immune function. The spleen is involved in immune surveillance and response, filtering blood to remove pathogens, antigens, and damaged or abnormal cells. It plays a key role in initiating immune responses against infections. Hematologic function. Red blood cell maintenance. The spleen helps regulate red blood cell production and turnover by removing old or damaged erythrocytes from circulation. Platelet storage. Acts as a reservoir for platelets, releasing them into circulation in response to bleeding or other physiological needs. Disease detection and diagnosis. Splenomegaly, enlargement of the spleen, often due to underlying conditions such as infections e.g., mononucleosis, liver diseases e.g., cirrhosis, hematologic disorders e.g., leukemia, lymphoma, and inflammatory conditions e.g., autoimmune diseases. Hypersplenism, condition where the spleen becomes overactive, leading to increased destruction of blood cells, hemolysis, and resulting in anemia, leukopenia, and thrombocytopenia. Splenectomy, surgical removal of the spleen, which may be necessary in cases of trauma e.g., splenic rupture, certain hematologic disorders e.g., hereditary spherocytosis, splenic tumors, or to control complications of splenomegaly. However, splenectomy increases the risk of infections, particularly those caused by encapsulated bacteria, due to loss of the spleen's immune functions. Diseases involving the spleen, infections. The spleen helps combat infections by filtering blood and initiating immune responses. However, certain infections, such as malaria and bacterial infections e.g., sepsis, can affect the spleen directly or lead to splenic complications. Hematologic disorders. Hemolytic anemias, conditions where red blood cells are prematurely destroyed, leading to anemia. The spleen's role in filtering and removing abnormal red blood cells can be exaggerated in these disorders. Thrombocytopenia, low. Clinical significance, immune function. The spleen is involved in immune surveillance and response, filtering blood to remove pathogens, antigens, and damaged or abnormal cells. It plays a key role in initiating immune responses against infections. Hematologic function. Red blood cell maintenance. The spleen helps regulate red blood cell production and turnover by removing old or damaged erythrocytes from circulation. Platelet storage. Acts as a reservoir for platelets, releasing them into circulation in response to bleeding or other physiological needs. Disease detection and diagnosis. Splenomegaly, enlargement of the spleen, often due to underlying conditions such as infections e.g., mononucleosis, liver diseases e.g., cirrhosis, hematologic disorders e.g., leukemia, lymphoma, and inflammatory conditions e.g., autoimmune diseases. Hypersplenism, condition where the spleen becomes overactive, leading to increased destruction of blood cells, hemolysis, and resulting in anemia, leukopenia, and thrombocytopenia. Splenectomy, surgical removal of the spleen, which may be necessary in cases of trauma e.g., splenic rupture, certain hematologic disorders e.g., hereditary spherocytosis, splenic tumors, or to control complications of splenomegaly. However, splenectomy increases the risk of infections, particularly those caused by encapsulated bacteria, due to loss of the spleen's immune functions. Diseases involving the spleen, infections. The spleen helps combat infections by filtering blood and initiating immune responses. However, certain infections, such as malaria and bacterial infections e.g., sepsis, can affect the spleen directly or lead to splenic complications. Hematologic disorders. Hemolytic anemias, conditions where red blood cells are prematurely destroyed, leading to anemia. The spleen's role in filtering and removing abnormal red blood cells can be exaggerated in these disorders. Thrombocytopenia, low platelet count, which may occur due to increased sequestration and destruction of platelets in an enlarged spleen. Hypersplenism. Cancers. Lymphoma, cancer affecting lymphocytes, which can involve the spleen directly or spread to it from other lymphatic tissues. Metastatic cancer, solid tumors e.g., breast, lung, melanoma can metastasize to the spleen, impacting its function and potentially causing splenic enlargement. Autoimmune diseases. Conditions such as systemic lupus erythematosus (SLE), rheumatoid arthritis, and immune thrombocytopenic purpura (ITP) can lead to splenomegaly and altered spleen function due to chronic immune activation and inflammation. Diagnostic and therapeutic interventions. Imaging, ultrasonography, CT scans, or MRI can help visualize the spleen and assess its size and structure. Splenectomy, surgical removal of the spleen may be necessary to treat underlying conditions or complications such as trauma, certain cancers, or severe splenomegaly. Vaccination. Patients undergoing splenectomy or with functional asplenia, absence of spleen function, are recommended to receive vaccinations against encapsulated bacteria e.g., pneumococcus, meningococcus, Haemophilus influenzae to reduce the risk of infections. Understanding the clinical significance of the spleen is essential for healthcare providers in diagnosing, managing, and treating various disorders that affect this vital organ's function and immune response, blood filtration, and hematologic balance. ELISA technique principle essay. Only about principle. I will ask you next things right away. ChatGPT. The enzyme-linked immunosorbent assay (ELISA) is a widely used technique in immunology and molecular biology for detecting and quantifying specific proteins, antibodies, antigens, and other molecules in biological samples. Here's an essay on the principle of ELISA. Principle of ELISA. The principle of ELISA revolves around the specificity of antigen-antibody interactions in the enzyme-linked detection system. The assay utilizes a solid-phase enzyme immunoassay to detect the presence and quantity of a target molecule in a sample. There are several types of ELISA, including direct, indirect, sandwich, and competitive formats, each tailored to different analytical needs. Solid-phase immunoassay. ELISA typically utilizes a microtiter plate as the solid phase, which is pre-coated with either capture antibodies in sandwich ELISA or antigens in indirect or direct ELISA. The solid phase facilitates the immobilization of the target molecule or antibody, ensuring it remains bound during subsequent wash steps. Antigen-antibody binding. The first step in ELISA involves the binding of the target molecule antigen to specific antibodies immobilized on the microtiter plate. This interaction is highly specific, with antibodies recognizing and binding to particular epitopes on the antigen of interest. Detection system. After washing away unbound molecules, a secondary antibody conjugated with an enzyme, e.g., horseradish peroxidase, alkaline phosphatase, is added. This secondary antibody binds to a different epitope on the antigen, forming a sandwich complex in sandwich ELISA or binding directly to the prime. The first step in ELISA involves the binding of the target molecule antigen to specific antibodies immobilized on the microtiter plate. This interaction is highly specific, with antibodies recognizing and binding to particular epitopes on the antigen of interest. Detection system. After washing away unbound molecules, a secondary antibody conjugated with an enzyme, e.g., horseradish peroxidase, alkaline phosphatase, is added. This secondary antibody binds to a different epitope on the antigen, forming a sandwich complex in sandwich ELISA or binding directly to the primary antibody in indirect ELISA. The enzyme linked to the secondary antibody catalyzes a colorimetric or fluorescent reaction when substrate is added. The intensity of the color or fluorescence is proportional to the amount of antigen present in the sample. Colorimetric or fluorescent signal detection. After incubation with substrate, the enzyme catalyzes the conversion of the substrate into a detectable product, e.g., colored product for chromogenic substrates. The color change is measured spectrophotometrically at a specific wavelength, providing a quantitative measure of the amount of antigen present in the sample. Applications and advantages. Clinical diagnostics. ELISA is widely used in clinical laboratories for diagnosing infectious diseases, e.g., HIV, hepatitis, autoimmune disorders, e.g., rheumatoid arthritis, and monitoring therapeutic drug levels. Biomedical research. It is crucial in research settings for studying protein-protein interactions, biomarker discovery, and drug development. High sensitivity and specificity. ELISA offers high sensitivity and specificity due to the specificity of antibody-antigen interactions and the enzymatic amplification of signal, making it a robust tool for quantitative analysis. Conclusion. In conclusion, ELISA is a powerful and versatile technique based on the principles of antigen-antibody interactions and enzyme-linked detection. Its ability to detect and quantify specific molecules with high sensitivity and specificity has made it indispensable in various fields, from clinical diagnostics to biomedical research. The methodological variations of ELISA cater to different analytical needs, making it adaptable for diverse applications in biological and medical sciences. Parts of machine and requirements. ChatGPT. To clarify, are you asking about the components or parts of an ELISA machine and the requirements for conducting an ELISA assay? Yeah, ChatGPT. Parts of an ELISA machine: microplate reader, microplate holder. Platform where the microplate containing samples and reagents is placed during the assay. Optical system. Includes light source, typically a xenon lamp, and photodetectors, photodiodes, or photomultiplier tubes for measuring absorbance or fluorescence signals from the microplate wells. Filter or monochromator. Allows selection of specific wavelengths of light for absorbance or fluorescence detection, depending on the assay requirements. Temperature control. Maintains a stable temperature, usually around 37 degrees Celsius, for kinetic assays or to ensure enzyme activity in certain ELISA types, e.g., enzyme activity assays. Shaking mechanism. Enables thorough mixing of reagents and samples within the microplate wells to ensure uniform reaction conditions. Data acquisition and analysis software. Software interface for controlling the machine, collecting absorbance or fluorescence data from the microplate wells, and analyzing the results. Requirements for conducting an ELISA assay. Microplate. Typically made of plastic or glass with 96, 384, or 1536 wells for holding samples, standards, controls, and reagents. Coating and blocking buffer. Buffer solutions used to coat the microplate wells with capture antibodies in sandwich ELISA or antigens in direct or indirect ELISA and to block non-specific binding sites. Primary antibodies capture and detection. Capture antibodies specific to the target antigen in sandwich ELISA or detection antibodies labeled with an enzyme in indirect or sandwich ELISA for antigen detection. Secondary antibody. Enzyme conjugated antibody specific to the primary antibody, used in indirect or sandwich ELISA to amplify the signal. Substrate solution. Chromogenic or fluorogenic substrate for the enzyme, e.g., horseradish peroxidase, alkaline phosphatase linked to the secondary antibody, producing a color change or fluorescent signal upon enzymatic reaction. Washing buffer. Buffer solution used to wash unbound molecules from the microplate wells between incubation steps to reduce background noise and increase assay specificity. Plate sealer or cover. Prevents evaporation during incubation steps and ensures consistent reaction conditions. Microplate reader, ELISA machine. Instrument for measuring and quantifying the absorbance or fluorescent signals from the microplate wells. Pipettes and tips. To accurately dispense small volumes of samples, reagents, and standards into microplate wells. Safety equipment. Lab coat, gloves, and safety goggles to protect against exposure to potentially hazardous chemicals and biological materials. Workflow summary. Coating. Coat microplate wells with capture antibodies or antigens. Blocking. Block non-specific binding sites with blocking buffer. Incubation. Add samples, standards, and controls to microplate wells, followed by incubation to allow antigen-antibody binding. Washing. Wash wells to remove unbound substances. Detection. Add enzyme-linked detection antibodies or secondary antibodies, incubate, and wash. Substrate addition. Add substrate solution to produce a detectable signal. Signal measurement. Measure absorbance or fluorescence in the microplate reader. Data analysis. Analyze data using software to quantify the target molecule concentration based on the signal generated. These components and requirements are essential for performing ELISA assays accurately and reliably in laboratory settings, supporting various applications in research, diagnostics, and biotechnology. Procedure and mechanism. ChatGPT. Procedure of ELISA enzyme-linked immunosorbent assay coating. Objective. Microplate wells are coated with a capture antibody or antigen to immobilize the target molecule. Procedure. Dilute the capture antibody or antigen in coating buffer. Add the diluted solution to microplate wells. Incubate overnight or for several hours at room temperature or 4 degrees Celsius. Wash to remove unbound antibodies or antigens. Blocking. Objective. Block non-specific binding sites on the microplate surface. Procedure. Add blocking buffer e.g., BSA, milk to the wells. Incubate for 1 to 2 hours at room temperature or specified. Wash to remove excess blocking buffer. Sample and standard addition. Objective. Add samples containing the target antigen and standards of known concentrations for calibration. Procedure. Add diluted samples, standards, and controls to respective wells. Incubate for a specified time to allow antigen-antibody binding. Detection. Objective. Detect the presence of the target antigen using an enzyme-linked detection antibody. Procedure. Dilute the enzyme-linked detection antibody in assay buffer. Add to microplate wells. Incubate for a specified time to allow binding of the detection antibody to the antigen. Wash to remove unbound detection antibody. Substrate addition. Objective. Add substrate solution to generate a detectable signal. Procedure. Add chromogenic substrate e.g., TMB or fluorogenic substrate to each well. Incubate for a short period to allow the enzyme to catalyze the substrate reaction. Signal measurement. Add the diluted solution to microplate wells. Objective. Microplate wells are coated with a capture antibody or antigen to immobilize the target molecule. Procedure. Dilute the capture antibody or antigen encoding buffer. Add the diluted solution to microplate wells. Incubate overnight or for several hours at room temperature or 4 degrees Celsius. Wash to remove unbound antibodies or antigens. Blocking. Objective. Block non-specific binding sites on the microplate surface. Procedure. Add blocking buffer e.g., BSA, milk to the wells. Incubate. Dilute the capture antibody or antigen encoding. Procedure of ELISA enzyme-linked immunosorbent assay coating. Objective. Microplate wells are coated with a capture antibody or antigen to immobilize the target molecule. Procedure. Dilute the capture antibody or antigen encoding buffer. Add the diluted solution to microplate wells. Incubate overnight or for several hours at room temperature or 4 degrees Celsius. Wash to remove unbound antibodies or antigens. Blocking. Objective. Block non-specific binding sites on the microplate surface. Procedure. Add blocking buffer e.g., BSA, milk to the wells. Incubate for 1 to 2 hours at room temperature or specified. Wash to remove excess blocking buffer. Sample and standard addition. Objective. Add samples containing the target antigen and standards of known concentrations for calibration. Procedure. Add diluted samples, standards, and controls to respective wells. Incubate for a specified time to allow antigen-antibody binding. Detection. Objective. Detect the presence of the target antigen using an enzyme-linked detection antibody. Procedure. Dilute the enzyme-linked detection antibody in assay buffer. Add to microplate wells. Incubate for a specified time to allow binding of the detection antibody to the antigen. Wash to remove unbound detection antibody. Substrate addition. Objective. Add substrate solution to generate a detectable signal. Procedure. Add chromogenic substrate e.g., TMB or fluorogenic substrate to each well. Incubate for a short period to allow the enzyme to catalyze the substrate reaction. Signal measurement. Objective. Measure the absorbance or fluorescence signal generated by the enzyme substrate reaction. Procedure. Use a microplate reader to measure absorbance at a specific wavelength for chromogenic substrates or fluorescence intensity for fluorogenic substrates. Record the optical density OD or relative fluorescence units RFU for each well. Data analysis. Objective. Analyze the data to determine the concentration of the target antigen in the samples. Procedure. Construct a standard curve using the absorbance or fluorescence readings of the standards with known concentrations. Calculate the concentration of the target antigen in the samples based on their respective absorbance or fluorescence values relative to the standard curve. Mechanism of ELISA. Antigen-antibody interaction. The key principle of ELISA relies on the specific binding between an antigen target molecule and an antibody that recognizes it. This interaction forms the basis for detecting and quantifying the antigen of interest. Enzyme-linked detection. In indirect and sandwich ELISA formats, after the antigen-antibody binding step, a secondary antibody linked to an enzyme e.g., horseradish peroxidase, alkaline phosphatase is added. This secondary antibody binds to a different epitope on the antigen or primary antibody, forming a complex. Substrate conversion. The enzyme catalyzes the conversion of a colorless substrate chromogenic or non-fluorescent substrate fluorogenic into a detectable product. For chromogenic substrates, this results in a color change visible to the naked eye or measurable with a microplate reader. For fluorogenic substrates, the enzyme reaction generates fluorescent molecules. Signal detection. The absorbance for chromogenic substrates or fluorescence intensity for fluorogenic substrates directly correlates with the amount of antigen present in the sample. The signal intensity is proportional to the concentration of the antigen, allowing for quantitative analysis. ELISA's versatility and sensitivity make it a widely used technique in biomedical research, clinical diagnostics, and biotechnology for detecting and quantifying various molecules, including proteins, antibodies, antigens, hormones, and pathogens. Uses, applications, and advantages. ChatGPT. Uses and applications of ELISA enzyme-linked immunosorbent assay. Uses. Clinical diagnostics. Detection of infectious diseases. ELISA is used to detect viral infections e.g., HIV, hepatitis, bacterial infections e.g., Lyme disease, and parasitic infections e.g., malaria. Autoimmune disease diagnosis. Helps in diagnosing autoimmune disorders e.g., rheumatoid arthritis, systemic lupus erythematosus by detecting autoantibodies. Hormone assays. Measures hormone levels e.g., thyroid hormones, cortisol for diagnosing endocrine disorders. Biomedical research. Biomarker discovery. Identifies and quantifies specific biomarkers associated with diseases or physiological processes. Drug development. Screens for potential drug targets and evaluates drug efficacy by measuring biomarker responses. Environmental monitoring. Detects pollutants and contaminants in water, soil, and food samples e.g., pesticides, toxins using antibody-based assays. Food safety testing. Screens for allergens e.g., gluten, peanuts, and contaminants e.g., toxins, pathogens in food products to ensure safety and regulatory compliance. Quality control in biotechnology. Monitors protein production and purification processes in biotechnology and pharmaceutical industries. Advantages. High sensitivity and specificity. ELISA offers high sensitivity, capable of detecting low concentrations of antigens or antibodies in biological samples. Specific binding between antibodies and antigens ensures accurate detection and minimal cross-reactivity. Quantitative results. Provides quantitative measurements of target molecules, allowing for precise determination of concentrations in samples. Versatility. Various ELISA formats e.g., direct, indirect, sandwich, competitive accommodate different assay requirements and analytes. Adapted for detecting proteins, antibodies, antigens, hormones, and other biomolecules. Speed and throughput. Can process multiple samples simultaneously using microplate formats, increasing assay throughput and efficiency. Ease of use and automation. Well-established protocols and availability of automated systems simplify assay setup and data analysis. Suitable for both manual and automated operation in laboratory settings. Cost effectiveness. ELISA kits are commercially available for many applications, reducing the need for specialized reagent preparation. Economical compared to other analytical techniques like PCR or mass spectrometry for certain applications. Conclusion. ELISA remains a cornerstone technique in biomedical sciences and diagnostics due to its reliability, sensitivity, and versatility. Its applications range from clinical diagnostics and biomedical research to environmental monitoring and food safety testing. Advancements in assay development and automation continue to enhance its utility in diverse fields, contributing to improved disease diagnosis, biomarker discovery, and quality assurance in various industries. Agarose gel electrophoresis principle. ChatGPT. Principle of agarose gel electrophoresis. Agarose gel electrophoresis is a fundamental technique used in molecular biology and biochemistry to separate and analyze nucleic acids, DNA, and RNA based on their size. This method exploits the migration of charged molecules through a gel matrix under the influence of an electric field. Here's an essay detailing the principle of agarose gel electrophoresis. Gel preparation. Agarose gel matrix. Agarose, a polysaccharide derived from seaweed, is the most common matrix used in gel electrophoresis. Its porous structure allows molecules to migrate through it based on size. Buffer solution. The gel is submerged in a buffer solution that conducts electricity and maintains pH stability during electrophoresis. Common buffers include tris acetate EDTA TAE or tris borate EDTA TBE. Staining agent. DNA or RNA molecules are typically visual. The gel is submerged in a buffer solution that conducts electricity and maintains pH stability during electrophoresis. Common buffers include tris acetate EDTA TAE or tris. Agarose, a polysaccharide derived from seaweed, is the most common matrix used in gel electrophoresis. Its porous structure allows molecules to migrate through it based on size. Buffer solution. The gel is submerged in a buffer solution that conducts electricity and maintains pH stability during electrophoresis. Common buffers include tris acetate EDTA TAE or tris borate EDTA TBE. Staining agent. DNA or RNA molecules are typically. The gel is submerged in a buffer solution that conducts electricity and maintains pH stability during electrophoresis. Common buffers include tris acetate EDTA TAE or tris borate EDTA TBE. Staining agent. DNA or RNA molecules are typically visualized using a fluorescent dye, e.g., ethidium bromide, or a non-carcinogenic alternative, e.g., SYBR-Safe. This dye intercalates between nucleic acid bases, allowing visualization under UV light. Electrophoresis process. Loading the gel. Samples containing nucleic acid. DNA or RNA molecules are typically visualized using a fluorescent dye, e.g., ethidium bromide, or a non-carcinogenic alternative, e.g., SYBR-Safe. This dye intercalates between nucleic acid bases, allowing visualization under UV light. Electrophoresis process. Loading the gel. Samples containing nucleic acids are mixed with loading buffer containing tracking dyes for visualization and density agents to ensure samples sink into wells before being loaded into wells at one end of the gel. Application of electric field. An electric current is applied across the gel through electrodes at both ends. DNA and RNA molecules, negatively charged due to their phosphate backbone, migrate towards the positive electrode anode. Separation based on size. During electrophoresis, smaller nucleic acid molecules move more quickly through the gel matrix than larger ones. This separation occurs because smaller molecules encounter less resistance from the gel matrix pores, allowing them to migrate further in a given period. Visualize. During electrophoresis, smaller nucleic acid molecules move more quickly through the gel matrix than larger ones. This separation occurs because smaller molecules encounter less resistance from the gel matrix pores, allowing them to migrate further in a given period. Visualization. After electrophoresis, the gel is stained with a dye that binds to nucleic acids. Under UV light, the stained bands of DNA or RNA appear as distinct bands on the gel, with each band representing a different size molecule. Applications of agarose gel electrophoresis. DNA fragment analysis, used to analyze PCR products, restriction digests, and genomic DNA for genetic research and diagnostics. RNA analysis, separates RNA molecules for gene expression studies, RNA interference, RNAi validation, and RNA quantification. Mutation detection, identifies genetic. Mutation detection, identifies genetic mutations and polymorphisms by comparing fragment sizes between individuals or samples. Microbial identification, analyzes microbial communities through gel-based fingerprinting techniques like amplified fragment length polymorphism, AFLP, or random amplified polymorphic DNA, RAPD. Advantages of agarose gel electrophoresis, simplicity, straightforward setup and operation with minimal equipment requirements. High resolution, resolves nucleic acids differing in size by just a few base pairs. Versatility, adaptable to various nucleic acid types, DNA, RNA, and sizes from small oligonucleotides to large genomic fragments. Cost effectiveness, affordable materials and reagents compared to other separation techniques like capillary electrophoresis or sequencing methods. Conclusion. Agarose gel electrophoresis remains indispensable in molecular biology laboratories for separating and analyzing nucleic acids based on size. Its principle of mobility through a gel matrix under an electric field enables researchers to study genetic diversity, mutations, and gene expression patterns crucial for understanding biological processes and diagnosing genetic disorders. Continuous advancements in staining methods and gel imaging technologies further enhance the utility and sensitivity of this technique in modern biotechnology and biomedical research. Agarose gel electrophoresis apparatus parts and requirements. ChatGPT. Agarose gel electrophoresis apparatus parts and requirements. Agarose gel electrophoresis requires specific equipment to separate and analyze nucleic acids based on size. Agarose gel electrophoresis requires specific equipment to separate and analyze nucleic acids based on size. Here are the essential parts and requirements for an agarose gel electrophoresis apparatus. Parts of the apparatus: electrophoresis tank, a plastic or acrylic tank that holds the agarose gel submerged in a buffer solution during electrophoresis. Power supply provides the electric current necessary for driving the migration of nucleic acids through the gel matrix. Typically provides voltages ranging from 50 volts to 300 volts depending on the gel size and desired separation resolution. Electrodes: platinum or graphite electrodes connected to the power supply. One electrode cathode is connected to the negative terminal, and the other anode to the positive terminal. These electrodes establish the electric field across the gel. Safety cover: transparent litter cover that protects the gel and buffer from contamination and minimizes buffer evaporation during electrophoresis. Gel casting tray holds the molten agarose solution during gel casting and solidification. It typically has cones or well formers to create sample wells for loading nucleic acid samples. UV transilluminator used for visualizing nucleic acid bands post electrophoresis. It emits UV light, typically 302 or 365 nanometers, to excite fluorescent dyes, e.g., ethidium bromide bound to nucleic acids in the gel. Gel imaging system, optional but highly useful for capturing digital images of the agarose gel after electrophoresis. Optional but highly useful for capturing digital images of the agarose gel after electrophoresis. Includes a camera, filter, and software for documentation and analysis of gel images. Optional but highly useful for capturing digital images of the agarose gel after electrophoresis. Includes a camera, filter, and software for documentation and analysis of gel images. Requirements for agarose gel electrophoresis. Agarose powder. High quality agarose with suitable gel strength and low electroendosmosis, EO, for optimal separation of nucleic acids. Buffer solution. Electrophoresis buffer such as trisacetate EDTA TAE or tris-4 EDTA TBE to maintain pH stability and provide ions for conductivity during electrophoresis. Staining agent. Fluorescent dye e.g. ethidium bromide, SYBR safe for visualizing nucleic acid bands under UV light. Ensure compatibility with the UV transilluminator wavelength. Loading dye. High quality agros with suitable gel strength and low electroendosmosis EO for optimal separation of nucleic acids. Buffer solution. Electrophoresis buffer such as trisacetate EDTA TAE or tris-4 EDTA TBE to maintain pH stability and provide ions for conductivity during electrophoresis. Staining agent. Fluorescent dye e.g. ethidium bromide, SYBR safe for visualizing nucleic acid bands under UV light. Ensure compatibility with the UV transilluminator wavelength. Loading dye. A denser solution added to nucleic acid samples to help them sink into wells and track migration during electrophoresis. Contains tracking dyes e.g. bromophenyl blue, xylene cyanol FF for visualization. Electrophoresis power supply. Provides consistent voltage and current for efficient separation of nucleic acids based on size. Ensure compatibility with the electrophoresis tank and electrodes. Pipettes and tips. For accurate dispensing of agros, buffers, samples, and loading dyes into gel casting trays and sample wells. Safety equipment. Personal protective equipment PPE such as gloves and safety goggles when handling agros, staining agents, and UV light sources to protect against chemical and UV exposure. Additional considerations. Gel documentation system for permanent documentation and analysis of gel images. Temperature control. Some systems offer cooling options to prevent gel overheating during electrophoresis, which can affect separation resolution. Maintenance and cleaning. Regular cleaning of electrodes and tanks to prevent contamination and ensure consistent performance. Agros gel electrophoresis apparatus and requirements are essential for performing nucleic acid separation and analysis in molecular biology laboratories. Proper setup and adherence to safety protocols ensure accurate and reliable results for various applications, including DNA fragment analysis, RNA analysis, and mutation detection. Procedure and mechanism ChatGPT. Procedure and mechanism of agros gel electrophoresis. Agros gel electrophoresis is a widely used technique in molecular biology to separate nucleic acids, DNA, or RNA based on their size. This method utilizes an agros gel matrix and an electric field to facilitate the migration of charged nucleic acid molecules through the gel. Here's a detailed overview of the procedure and mechanism involved. Procedure: preparing the gel. Gel preparation. Prepare an agros solution by dissolving agros powder in an appropriate buffer, e.g., TAE or TBE buffer. Heat the mixture to dissolve agros completely, then allow it to cool to about 50-60 degrees Celsius before pouring into a gel casting tray. Creating wells. Insert a comb into the gel casting tray to create wells where samples will be loaded. The number of wells and their placement depend on the gel size and the number of samples. Solidification. Allow the agros solution to solidify at room temperature. Once solidified, carefully remove the comb to leave wells for loading samples. Preparing samples. Mixing with loading dye. Mix nucleic acid samples with a loading dye containing tracking dyes, e.g., bromophenyl blue, xylene cyanol FF, and density agents to ensure samples sink into wells. Denaturation if needed. For DNA, heat denature the sample. Mixing with loading dye. Mix nucleic. Allow the agros solution to solidify at room temperature. Heat the mixture to dissolve agros completely, then allow it to cool to about 50-60 degrees Celsius before pouring into a gel casting tray. Creating wells. Insert a comb into the gel casting tray to create wells where samples will be loaded. The number of wells and their placement depend on the gel size and the number of samples. Solidification. Allow the agros solution to solidify at room temperature. Once solidified, carefully remove the comb to leave wells for loading samples. Preparing samples. Mixing with loading dye. Mix nucleic acid samples with a loading dye containing tracking dyes, e.g., bromophenyl blue, xylene cyanol FF, and density agents to ensure samples sink into wells. Denaturation if needed. For DNA, heat denature the samples if single-stranded DNA is required, typically by heating at 95 degrees Celsius for 5 minutes. Loading the gel. Loading samples. Carefully load the nucleic acid samples mixed with loading dye into the wells using a micropipette. Ensure equal volumes are loaded for accurate comparison. Electrophoresis. Setting up the apparatus. Place the gel casting tray into the electrophoresis tank filled with electrophoresis buffer, e.g., TAE or TBE, that covers the gel completely. Applying voltage. Connect the electrodes anode and cathode of the power supply to the electrophoresis tank. Apply a constant voltage, typically 50 to 150 V depending on the gel concentration and desired resolution, across the gel for a set period, usually 30 minutes to several hours, to drive nucleic acids through the gel matrix. Visualization. Staining the gel. After electrophoresis, stain the gel with a nucleic acid stain, e.g., ethidium bromide, SYBR safe, that binds to DNA or RNA molecules. Allow the stain to penetrate the gel for about 15 to 30 minutes. Visualization under UV light. Place the gel on a UV transilluminator, which emits UV light, typically 302 or 365 nanometers. The stained nucleic acid bands fluoresce under UV light and can be visualized and photographed. Analysis. Interpreting results. Compare the migration distances measured as bands of nucleic acid samples with molecular weight markers, DNA ladders of known sizes. Analyze the gel image to determine the size and quantity of nucleic acid fragments present in each sample. Mechanism. Gel matrix. Agros gel acts as a sieve, separating nucleic acid molecules based on size. Smaller molecules move more easily through the gel matrix pores than larger molecules. Electric field. When an electric current is applied, nucleic acids, which have negatively charged phosphate groups, migrate towards the positive electrode anode through the agros gel. Separation by size. During electrophoresis, smaller nucleic acid fragments travel faster and migrate further through the gel compared to larger fragments. This separation results in distinct bands that represent different size DNA or RNA molecules. Visualization. Staining with a nucleic acid dye allows the visualization of nucleic acid bands under UV light. Each band corresponds to a different size fragment of DNA or RNA present in the sample. Agros gel electrophoresis is crucial for various applications in molecular biology, including DNA fragment analysis, RNA analysis, mutation detection, and genetic research. Its simplicity, versatility, and ability to provide accurate size separation of nucleic acids make it a cornerstone technique in biological and biomedical sciences. Uses, applications, and advantages. ChatGPT. Uses, applications, and advantages of agros gel electrophoresis. Agros gel electrophoresis is a fundamental technique widely employed in molecular biology and biochemistry for separating nucleic acids, DNA, and RNA based on their size. This method leverages the migration of charged molecules through a gel matrix under the influence of an electric field. Here's an overview of its uses, applications, and advantages. Uses and applications. DNA fragment analysis. Purpose: determines the size distribution of DNA fragments produced by restriction enzyme digestion, PCR amplification, or cloning. Application: essential for genetic research, mapping DNA sequences, and analyzing gene structure and function. RNA analysis. Purpose: separates RNA molecules by size for studying gene expression, RNA interference, RNAi, and RNA processing. Application: crucial in gene expression profiling, RNA sequencing, RNA-seq, and microRNA analysis. Mutation detection. Purpose: identifies genetic mutations, polymorphisms, or sequence variations in DNA samples. Application: used in clinical diagnostics, genetic counseling, and disease susceptibility studies. Microbial fingerprinting. Purpose: analyzes genetic diversity and relatedness among microbial strains or communities. Application: important in epidemiological studies, microbial ecology, and monitoring microbial populations in environmental samples. PCR product verification. Purpose: confirms the success of PCR amplification and verifies the size of PCR products. Application: ensures accurate downstream applications such as cloning, sequencing, and genetic screening. Genetic engineering and biotechnology. Purpose: characterizes DNA constructs, plasmids, and recombinant proteins. Application: supports biotechnological advancements, including gene editing, CRISPR, SOS9 synthetic biology, and vaccine development. Forensic science. Purpose: analyzes DNA evidence to identify suspects, victims, or genetic relationships in forensic investigations. Application: provides critical evidence in criminal cases, paternity testing, and disaster victim identification. Advantages: high resolution. Separates nucleic acids with high resolution, allowing detection of small differences in fragment sizes. Ease of use. Simple setup and operation make it accessible to researchers and technicians with basic training. Versatility. Applicable to various nucleic acid types, DNA, RNA, and sizes, from small oligonucleotides to large genomic fragments. Quantitative analysis. Allows for quantitative determination of nucleic acid concentrations based on band intensity in stained gels. Cost effectiveness. Requires relatively inexpensive materials, e.g., agarose, electrophoresis buffer, staining dyes compared to alternative methods like capillary electrophoresis or sequencing. Speed. Rapid separation and visualization of nucleic acids within a few hours, depending on gel size and electrophoresis conditions. Visualization. Bands of separated nucleic acids can be easily visualized and photographed under UV light, facilitating documentation and analysis. Agarose gel electrophoresis remains an indispensable tool in biological research, clinical diagnostics, forensic science, and biotechnology. Its robustness, reliability, and ability to provide detailed molecular insights continue to drive advancements in understanding genetic processes and disease mechanisms, as well as in developing innovative biotechnological applications. PCR principle ChatGPT. PCR principle polymerase chain reaction. PCR polymerase chain reaction is a widely used molecular biology technique that amplifies specific segments of DNA through repeated cycles of heating and cooling. Here's an overview of the principle behind PCR. Purpose. PCR allows researchers to make millions to billions of copies of a specific DNA sequence from a complex mixture, enabling detailed analysis, sequencing, and manipulation of DNA. Components involved: DNA template. The DNA sample containing the target sequence to be amplified. Primers. Short single-stranded DNA sequences, typically 18 to 22 nucleotides long, that are complementary to the sequences flanking the target DNA region. DNA polymerase. Enzyme that synthesizes new DNA strands complementary to the template DNA. Taq polymerase, derived from Thermus aquaticus, is commonly used due to its heat stability. DNTPs, deoxynucleotide triphosphates. Building blocks A, T, C, G used by DNA polymerase to synthesize new DNA strands. Buffer solution. Provides a suitable chemical environment, e.g., pH, ionic strength for DNA polymerase activity and stability. MgCl. Cofactor required by DNA polymerase for its enzymatic activity. Steps involved in PCR. Denaturation. Temperature, around 94-98 degrees Celsius. Purpose. Heat separates the double-stranded DNA template into two single strands. Denaturation, exposing the target sequence. Annealing. Temperature, typically 50-65 degrees Celsius. Purpose. Primers bind anneal to the complementary sequences flanking the target DNA region on each single-stranded template. Extension, elongation. Temperature, usually 72 degrees Celsius. Purpose. DNA polymerase synthesizes new DNA strands by extending from the primers along the template DNA using DNTPs. Cycling. The denaturation, annealing, and extension steps constitute one PCR cycle. Multiple cycles, usually 20 to 40 cycles, are performed to exponentially amplify the target DNA region. Amplification. Exponential growth. Each cycle doubles the number of DNA molecules, resulting in a logarithmic increase in the number of copies of the target sequence. End result. After PCR amplification, millions to billions of copies of the target DNA segment are produced from even minute amounts of starting material. These copies can be used for various downstream applications such as DNA sequencing, cloning, mutation analysis, and genetic testing. Applications. Research, molecular cloning, gene expression analysis, genetic mapping, and evolutionary studies. Medicine, diagnostic tests for genetic diseases, infectious diseases, and cancer. Forensics, DNA profiling for individual identification and criminal investigations. Biotechnology, production of recombinant proteins, genetic engineering, and agriculture. PCR's ability to amplify specific DNA sequences quickly, accurately, and with high sensitivity has revolutionized molecular biology, enabling numerous advances in research, diagnostics, and biotechnology. Apparatus parts, requirements ChatGPT. PCR apparatus, parts and requirements. PCR polymerase chain reaction is carried out using specialized equipment designed to perform the steps of DNA amplification efficiently and accurately. Here are the essential parts and requirements of a PCR apparatus. Parts of the apparatus, thermal cycler. The main unit of the PCR apparatus that controls the temperature cycles required for denaturation, annealing, and extension of DNA. It contains heating block, where the PCR tubes or plates are placed and subjected to precise temperature changes. Temperature control system, maintains and adjusts temperatures during different stages of the PCR process. PCR tubes or plates. Small reaction vessels where the PCR reaction mixtures are prepared and thermal cycled. These can be individual tubes or microplate format 96 well or 384 well plates for high throughput applications. Heating and cooling system. Rapidly heats and cools the samples to specific temperatures required for denaturation, annealing, and extension steps of PCR cycles. Lid or cover. Prevents condensation and evaporation of reaction mixtures during thermal cycling. Some lids also apply pressure to ensure proper sealing of PCR tubes or plates. User interface. Control panel or software interface for programming and monitoring the PCR run, setting temperature parameters, and viewing real-time thermal profiles. Gradient feed. Lid or cover. Rapidly heats and cools the samples to specific temperatures required for denaturation, annealing, and extension steps of PCR cycles. Lid or cover. Prevents condensation and evaporation of reaction mixtures during thermal cycling. Some lids also apply pressure to ensure proper sealing of PCR tubes or plates. User interface. Control panel or software interface for programming and monitoring the PCR run, setting temperature parameters, and viewing real-time thermal profiles. Gradient feature optional. Allows for testing multiple temperatures simultaneously across the thermal block to optimize annealing temperatures for different primer pairs. Requirements for PCR. DNA template. DNA sample containing the target sequence to be amplified. Primers. Short single-stranded DNA sequences specific to the sequences flanking the target DNA region. DNA polymerase. Thermostable DNA polymerase, such as Taq polymerase, capable of withstanding high temperatures during PCR cycles. DNTPs, deoxynucleotide triphosphates. Building blocks adenine, thymine, cytosine, guanine used by DNA polymerase to synthesize new DNA strands. Buffer solution. Provides optimal pH and ionic conditions for DNA polymerase activity and stability during PCR. MgCl. Cofactor required by DNA polymerase for enzymatic activity. PCR tubes or plates. Made from materials e.g., polypropylene that are compatible with thermal cycling and do not interfere with PCR reactions. PCR reagents and consumables. Includes molecular grade water, loading dyes, and molecular biology grade reagents for preparing PCR reaction mixtures. Disposable tips and pipettes. For accurate and contamination-free transfer of reagents and samples during PCR setup. PCR workflow setup. Dedicated work area for preparing PCR reactions, handling PCR products, and setting up reactions under sterile conditions to prevent contamination. Safety precautions. Personal protective equipment PPE such as gloves and lab coats to ensure safe handling of chemicals and biological materials used in PCR. Additional considerations. Quality control, regular calibration and maintenance of the thermal cycler to ensure accurate temperature control and reproducible PCR results. Data analysis software, software for analyzing PCR results, including gel electrophoresis images or real-time fluorescence data, depending on the PCR application. PCR apparatus and requirements are crucial for performing DNA amplification in various fields, including molecular biology research, clinical diagnostics, forensics, and biotechnology. Proper setup, adherence to protocols, and quality assurance measures ensure reliable and reproducible PCR results for diverse applications. Procedure and mechanism ChatGPT. PCR polymerase chain reaction procedure and mechanism. PCR polymerase chain reaction is a powerful molecular biology technique used to amplify specific segments of DNA through a series of temperature-controlled cycles. This process allows researchers to make millions to billions of copies of a particular DNA sequence from a complex mixture. Here's a detailed overview of the procedure and mechanism involved in PCR. Procedure. Setup. DNA template. Prepare a DNA sample containing the target sequence to be amplified. This can be genomic DNA, cDNA, or DNA obtained from various sources. Primers. Design and synthesize short single-stranded DNA oligonucleotides, typically 18 to 22 nucleotides that are complementary to the sequences flanking the target DNA region. Primers serve as starting points for DNA synthesis. PCR reaction mix. Prepare a PCR reaction mix containing DNA template. Forward and reverse primers DNA polymerase, e.g., Taq polymerase, DNTPs, deoxynucleotide triphosphates, A, T, C, G buffer solution providing optimal pH and ionic conditions, MgCl, cofactor required for DNA polymerase activity, PCR cycling. PCR consists of repeated cycles of three main steps: denaturation, annealing, and extension. The number of cycles typically ranges from 20 to 40, depending on the initial amount of DNA and the desired amount of amplification. Denaturation. Temperature, around 94-98 degrees Celsius. Time, 20 to 30 seconds. Purpose, heat melts the double-stranded DNA template into two single strands, breaking hydrogen bonds between complementary bases. Annealing. Temperature, typically 50-65 degrees Celsius, depends on primer melting temperature, TM time, 20 to 40 seconds. Purpose, primers bind anneal to the complementary sequences flanking the target DNA region on each single-stranded template. Extension, elongation. Temperature, usually 72 degrees Celsius. Time, 30 seconds to 2 minutes, depends on the length of the DNA fragment. Purpose, DNA polymerase synthesizes new DNA strands by extending from the primers along the template DNA, using DNTPs as building blocks. PCR amplification. Each cycle of denaturation, annealing, and extension doubles the number of DNA molecules, resulting in an exponential increase in the number of copies of the target DNA sequence. Final extension. After the last cycle, a final extension step at 72 degrees Celsius for 5 to 10 minutes ensures complete synthesis of all DNA strands. Mechanism. Denaturation. Heat breaks the hydrogen bonds between complementary bases, separating the double-stranded DNA into two single strands. Annealing. Primers anneal to the complementary sequences at the three feed ends of each single-stranded DNA template. Extension. DNA polymerase synthesizes new DNA strands by extending from the primers, using DNTPs that base pair with the template DNA. Cycle repetition. Repeated cycles of denaturation, annealing, and extension amplify the target DNA exponentially, producing millions to billions of copies within a short period. Applications. Genetic research. Study gene expression, genetic variation, and genomic sequencing. Clinical diagnostics. Detect genetic diseases, pathogens, and mutations. Forensics. Analyze DNA evidence for criminal investigations and paternity testing. Biotechnology. Produce recombinant DNA, clone genes, and engineer organisms. PCR speed, specificity, and sensitivity have revolutionized molecular biology and biotechnology, enabling numerous applications in research, medicine, and industry. Proper setup, optimization of reaction conditions, and stringent quality control ensure reliable and reproducible results in diverse PCR applications. Applications, uses, and advantages. ChatGPT. Applications, uses, and advantages of PCR polymerase chain reaction. PCR polymerase chain reaction is a versatile molecular biology technique that revolutionized the study of DNA by allowing researchers to amplify specific DNA sequences exponentially. Here's an overview of its applications, uses, and advantages. Applications, genetic research. Gene expression analysis, study gene regulation and expression patterns. Genomic sequencing, sequence specific regions of interest in genomes. Genetic mapping, map genes to specific chromosomes and loci. Clinical diagnostics. Infectious disease diagnosis, detect pathogens like viruses and bacteria. Genetic disease screening, identify mutations associated with inherited disorders. Cancer biomarker detection, analyze genetic markers related to cancer susceptibility and prognosis. Forensic science. DNA profiling, compare DNA samples for forensic identification purposes. Crime scene investigation, analyze trace DNA evidence left at crime scenes. Biotechnology. Recombinant DNA technology, clone genes and engineer DNA constructs. Drug development, validate drug targets and screen for genetic markers related to drug response. Environmental microbiology. Microbial ecology, study microbial diversity and community dynamics in ecosystems. Environmental monitoring, detect pathogens and contaminants in environmental samples. Ancient DNA studies. Paleogenetics, amplify and analyze DNA from ancient specimens for evolutionary studies. Agriculture. Crop improvement, identify and characterize genes related to agronomic traits. GMO detection, detect genetically modified organisms in food and agricultural products. Uses. DNA amplification, amplify specific DNA sequences from even minute amounts of starting material, facilitating downstream analysis. Mutation detection, identify genetic mutations, polymorphisms, and sequence variations associated with diseases and traits. Quantification, measure gene expression levels and viral load quantitatively. Sequencing template preparation, generate templates for DNA sequencing by amplifying regions of interest. Cloning and genetic engineering, clone genes, create libraries, and engineer DNA constructs for various biotechnological applications. Advantages. Sensitivity, detects low quantities of DNA, enhancing detection sensitivity for rare alleles or low abundance targets. Specificity, amplify specific DNA sequences, minimizing non-specific amplification. Speed, rapid amplification process allows results within hours compared to traditional cloning methods. Versatility, applicable to various DNA types, e.g., genomic DNA, cDNA, plasmid DNA, and targets, e.g., genes, microorganisms. High throughplut, adapted for high throughput applications using microplate formats and automation. Accuracy, provides precise and reproducible results with optimized reaction conditions. Cost effectiveness, requires minimal amounts of reagents and sample, reducing overall cost per reaction. PCR's ability to amplify DNA selectively and exponentially has transformed biological research, diagnostics, and biotechnology, enabling advancements in understanding genetics, disease mechanisms, and environmental microbiology. Continuous innovation in PCR technology and applications expands its utility in various scientific disciplines and industries.